Hello everyone. Today we are going to discuss affinity chromatography. Affinity chromatography, it's a very important technique in analytical chemistry which exploits the unique property of extremely specific biological interactions to achieve separation and purification. The technique was originally developed for the purification of enzymes, but now it has been extended for nucleotides, nucleic acids, immunoglobulins, membrane receptors, and even to whole cells. Now, when we say extremely specific biological interaction, we mean to talk about the specific interaction of a ligand for its analyte. Giving a simple example of, for example, every lock has a unique key. So similarly, these ligands, they have affinity for a particular type of analyte to which it binds. Therefore, the purification that is achieved with the help of affinity chromatography is of several thousand-fold, and the recovery of the material is very high. As you can see in this diagram, there is a matrix which we call as the stationary phase. To this, we attach a ligand which has a specific affinity for our target compound, which is actually mixed in a sample. So, when this ligand binds to this target molecule by a specific adsorption, it helps in the purification of this compound from a mixture that has a number of such molecules, but of course they don't have the affinity for this ligand. And this interaction between the ligand and the compound is reversible. It has to be reversible in order to purify this compound further. Now, this affinity chromatography is a unique purification technology, and it is the only technique that enables the purification of a biomolecule on the basis of its biological function, that is the biological properties or individual chemical structure. So separates proteins on the basis of reversible interaction. That is what I was talking about before, that the interaction between the ligand and the analyte has to be reversible. Now, this technique has three main steps. First is the incubation of the sample, the crude sample, which is actually a mixture with the affinity support, that is the matrix, to allow the target molecule in the sample to bind to the immobilized ligand. Means my target analyte will bind to the ligand in this step. Next is the washing step, in which the non-bound components of the sample, they are washed away from the matrix. Now, once the ligand has bound to the target analyte, the target analyte has to be finally recovered. So for this step, we call this step as elution step, in which there is a dissociation and recovery of the target analyte from the immobilized ligand, and this is usually done by changing the buffer conditions or the binding conditions between the target and the ligand. This is a diagrammatic representation which shows how the steps are done. In the very first stage, this is the matrix with immobilized ligand, and the sample is injected in the column. Specific adsorption of my target takes place with the ligand, and as you can see, the unbound components of the sample, they are still in the column. This is the washing step. The third is the washing step, in which the unbound, or you can say non-bound, sample components have been eluted from the column. Now, this is at this step, the adsorption between the target and the ligand has taken place, and the non-bound components has now left the column. The last is the elution step, in which we elute out the desired components, or the what you call at our target analyte. And this is done by either changing the buffer conditions, the pH, or competitive binding. That we'll be discussing in detail in the later stage. So what type of biological interactions play between the ligand and the target molecule? They can be electrostatic, they can be hydrophobic, they can be van der Waals forces, or they can be hydrogen bonding. For example, the interaction between the enzyme and the substrate, or the inhibitor or the cofactor, the antigen and the antibody, the lactins, which are special proteins for the polysaccharides, the nucleic acids for their complementary base sequences, nucleic acid polymerase enzymes. They can be hormones and vitamins. They have for the receptors, glutathione, we have metal ions. So these type of specific biological interactions are made use in affinity chromatography. Now, the matrix, it also plays a very important role. The matrix has to have certain features for proper selection, right? For proper running of affinity chromatography. It should be insoluble. It should be sufficiently permeable to allow the ligand to bind and the non-bound molecules and the bound molecules to flow through the matrix. It should be highly rigid and it should be suitable particle form. It should have zero adsorption capacity. It should be chemically stable. It should be chemically reactive to allow the ligand and the spacers to be introduced. The term spacers I will be introducing into the next few slides. Resistance towards the microbial and enzymatic attacks. It should have good flow properties for allowing the rapid separation, and the pore structure should ensure high capacity binding even for the large biomolecules. Although no matrix is perfectly ideal, but in affinity chromatography, generally used support materials are porous support materials like agarose, polymethyl polyacrylamide, cellulose, and silica. All the support materials, they are commercially available and they come in different particle and pore size range. Some supports may be available with common affinity ligands already immobilized. Other types of support materials have been developed, including non-porous supports, membranes, flow-through beads, monolithic supports, and expanded bead adsorbents. Next to the matrix is the next important thing is the ligand. Now, for a successful affinity purification, you need a specific ligand that is attached to the matrix covalently. The coupled ligand must retain its specific binding affinity for the target molecule, and after washing of the unbound material, the binding between the ligand and the target molecule must be reversible, so that the target molecule can be finally recovered from the column in the active form itself. This figure shows the matrix, and this is actually we require activation of the matrix. We do it by certain techniques. We activate the matrix so that the ligand can be attached to the matrix. Ligand immobilization is done by a number of methods, and this is done, for example, by activation of the first we have the activation step in which the matrix is activated, and then we have the finally the coupling step. In coupling step, this is, you can see how the matrix is linked with the ligand. There is one important thing, that is spacer arm. Now the binding sites of a target protein is often deeply located within the molecule and an affinity medium prepared. So coupling small ligands directly to the matrix may sometimes show steric interference, like there might not be proper space available for the ligands to bind, and once there is not enough space for the ligands to bind, there will not be enough space for the target analyte to interact properly with the ligand. So we make use of the spacer arms. So this figure shows how a ligand is binding to the target analyte, and in that case there is an insufficient binding of the target and because of which the illusions are not proper. But once we use these spacer arms, which are certain length of carbon sequences, this analyte properly interacts with the ligand and is also properly eluted, giving us a single peak. This is so once the absorption has taken place, now we come to the elution. The elution can be done, as we discussed before, by changing the buffer composition. We can change the pH or the salt concentrations, or we can go for competitive binding. Means we can introduce into a column a molecule that has better binding capacity or better interaction with the analyte, and so the specific elution will take place. Coming to the applications of affinity chromatography, we have lectin affinity chromatography. Lectins are basically proteins, non-immune system proteins such as glycoproteins, which show affinity for carbohydrates. So lectin affinity chromatography is between lectins and carbohydrates. Similarly, we have immobilized metal ion affinity. So immobilized metal ion affinity, this involves binding with target molecules like proteins, nucleic acids, amino acids with the metal ions. Similarly, we have dye-ligand affinity chromatography, in which we have columns that contains dyes which help in to remove molecules such as hepatitis B viral particles or prime proteins. Similarly, we have immunoaffinity, in which we make use of antibodies that binds to various peptides, hormones, enzymes that can be purified. So this is all about affinity chromatography. Thank you.